Hello everyone, this is Alex, Nate, Max, and Trenton's green chemistry presentation on cycloalkene catalysis using whole cell enzymes and renewable substrates. We will focus on a process called enzymatic cascade, which is defined as a chemical process in which the product of each preceding reaction catalyzes and is consumed in the next reaction. In this example, E. coli is used as a vector for these reactions. E. coli contains the necessary enzymes for the cascades to take place. E. coli is a great mechanism because it is a common bacteria used in scientific research and its metabolic pathway is well known. Additionally, cycloalkenes, aromatics, and linear alkenes are produced using oil and sugars as the renewable substrates in the reactions. It is done using a one-pot biocatalytic process. The process is defined as the chemical conversions of organic compounds using enzymes that still reside inside living cells. Man-made enzymatic cascades allow humans to convert renewable resources such as oils and sugars into desirable products such as cycloalkenes, as seen in the last slide. Cycloalkenes are useful products because they are bulk petrochemicals widely employed in various industrial processes. They are the building blocks that are essential to making the materials necessary for many aspects of modern life, like plastics, spacesuits, and paints. Originally, the process took place using adipic acid, a highly relevant commodity that is produced at large scale. However, the product is mainly obtained by an inefficient and environmentally harmful two-step process, and it is the largest source of industrial greenhouse gas emissions. The adipic acid procedure is incredibly inefficient at a conversion rate of less than 10%. It is important to note that the green chemistry aspect of this experiment. Previous types of chemistry release toxic chemicals into the atmosphere. However, to include a more green aspect of the procedure, chemicals are now either reused or used more efficiently. In this case, the chemicals are used more efficiently in a single natural system that already exists, rather than contaminating the planet with another non-biodegradable system or chemical. It also reduces the number of steps in the reaction and uses renewable substrates. By using greener chemistry, plants and animals will suffer less from the toxic chemicals being released, and it is a way to combat climate change, which has been a rising global concern in the scientific community.
In a 2002 UN World Summit on Sustainable Development held in Johannesburg, it was shown that enough biomass could be produced so that food supply and industrial demands would be met. Previously, there was concern that increasing the amount of biomass used for industrial and synthetic purposes would have disastrous effects on global food demands. However, this is simply not the case. The 2002 summit was called for the purpose of promoting the use of biomass in place of non-renewable resources and further greening the production of important fuel sources such as biodiesel. According to a 2011 paper by Dr. Ursula Biermann, a food chemistry researcher at Hanover and Munich studying thermal addition reactions in unsaturated fatty compounds, there have been promising advances made in the last decade in synthesizing double-bonded and ring structures from plant-derived oils and oleochemicals. Biermann even claims that oils and fats of vegetable and plant origin are historically and currently the most important renewable feedstock of the chemical industry. In the rightmost figure, you can see examples of popular oleochemicals, which are precursor compounds derived from animal fats or vegetable oils. With global production of such chemical compounds increasing each year, Biermann proposes that organic chemists and geneticists alike focus their efforts on finding greener alternatives to current industrial practices via the use of renewable oils. This way, we can take advantage of the skyrocketing production of these oils. The leftmost image on this slide compares the global production of oils in the 1999 to 2000 production year to the 2009 to 2010 production year. Here we can see there was an increase in all oil types observed, and notably we see a more than 100% increase in palm and palm kernel oil production. From 2001 to 2008, the global supply of fatty acids has nearly doubled. However, production and consumption have only slightly increased. The Biermann paper makes specific mention of a number of enzymatic and microbial transformations to take advantage of the global excess in oleochemicals. Notably, the use of certain enzymes that can be recombinantly expressed in E. coli, offering a greener alternative to the production of aromatics as seen with P450 monooxygenases, which is used for microbial hydroxylation of fatty acids.
From the data and information that Max and Alex have provided, it is abundantly clear that the production of linear alkanes and alkenes, aromatics, and cycloalkenes will not be slowing down, and in fact has been picking up. Thus, it is important to understand this reaction and its mechanisms in order to continue to implement and expand on it further. As stated before, This reaction begins with renewable reagents such as oils and sugars. These reagents are put into the one-pot reaction apparatus, which in this case is E. coli whole cells, to be subjected to enzymatic pathways, such as the hydrolysis of olive oil to oleic acid, the main reagent utilized in this reaction by Wu et al. The mechanisms as shown in step C yield short-lived intermediates that can be seen in step D, prior to being exposed to another slew of enzymes, such as the decarboxylase enzyme UNDB, that is a membrane-bound desaturase-like enzyme from Pseudomonas in step E, that are used to trim down and or form the intermediates into the final products. One special note, however, is that the Ru ruthenium-2 catalyst is utilized to close the rings of straight-chain hydrocarbon chains to form cycloalkenes, as opposed to removing a certain functional group, as other enzymes do. This catalyst is key in forming cycloalkenes in a green way.
Before the use of fatty acids, such as oleic acid, fossil fuels were used to produce cycloalkenes. One such example is the synthesis of cyclopentadiene and cyclopentene via pyrolysis of gasoline. The figure on the slide shows the reaction steps for the production of cyclopentene from crude dicyclopentadiene derived from coal tar or steam cracking operations. According to Martin Klaus and colleagues in an article outlining prior methods of cyclopentadiene and cyclopentene synthesis, this process requires high temperatures and high heat and releases residual gas as a non-green byproduct. In this process, crude dicyclopentadiene is monomerized in the cracking column at 170 to 190 degrees Celsius. The problem, however, with using crude dicyclopentadiene as a starting material is that it is highly toxic, as 100% of rats exposed to saturated dicyclopentadiene vapor for 1 hour experience treatment-related mortality. In humans, dicyclopentadiene has been shown to cause irritation of the eyes and respiratory tract in concentrations as low as 1 part per million. In general, green routes of forming cyclic compounds are shown to be safer and more cost-effective.
Some strengths are that this new process takes place as a single pot reaction, which are very efficient. This leads to less intermediate steps, and by having less intermediate steps, the reaction can really cut down on the time in which it takes for the reaction to reach completeness. The lack of intermediate steps also leads to less waste produced by the reaction. By using this new, greener procedure, there is also an increase in yield. Some limitations are that there is still room for more active enzymes to be used in this process. Chemoenzymatic cascades, reported to date, are limited, and there's only 1 or 2 enzymatic steps. We believe that this process can be... Many enzymes used in this reaction are also very substrate specific, therefore they cannot be implemented widely. Another limitation is that these enzymes have been tested, but only strong evidence has came about during the testing within E. coli. Overall, there is a lot of unknowns in the process that we have described above, and this new greener reaction may be more efficient, but is also mostly unknown on how to improve and make more efficient. In chemistry, a one-pot synthesis reaction has been used as a strategy to improve the efficiency of the chemical reaction, whereby a reactant is subjected to successive chemical reactions in just one reactor. There has also been a lot of exploration into decarboxyl and decarbonylase enzymes in the past. Previous works also looked to synthesize cycloalkenes, but these projected processes were going to have right around 9 steps. The Ru catalyst has been discussed by Robert Grubbs in 2009 to a great extent, and has actually provided a lot of research into this new field. Cycloalkane synthesis has been discussed as a possibility by scientists like Ilya Pavlov in 2016, but has only been recently attempted.
In conclusion, the implementation of green chemistry, and this reaction in particular, into commercial practice will cut down on greenhouse gases such as nitrous oxide, as well as the fossil fuel reagents and heavy metal catalysts that produce the gas and are very toxic in and of themselves. As for future works, we believe that Wu's suggestion of directed evolution of less substrate-specific enzymes will be the next step in manufacturing a more widespread process. We also believe that attempting identical or similar reactions in more strains and types of cells with greater variety of substrates would also assist in determining the most efficient mechanism for this reaction. This reaction is just the tip of the needle when it comes to the potential of green chemistry in the commercial field, and in the opinion of this group, there is nowhere better to start than here.